Hello everyone, welcome to Plant Biotechnology. This lecture is going to be Introduction to Plant Biotechnology, and here we have the lecture outlines. The first one, methods of biotechnology, gene transfer, plasmids, restriction enzymes, polymerase chain reaction, DNA ligase, and cloning. So these are the outlines we have it in plant biotechnology. If you see methods of biotechnology, so these methods will try to accomplishment and opportunities which is lies in plant biotechnology. And gene transfer methods, which is basically genetic engineering tool. And whereas plasmids, this will have transgenic plant in nature. And if you see the restriction enzymes, so which has both advantage and disadvantage in genetic DNA modifications. If you see the polymerase chain reactions, where we will have the engineering related field. And also if you see the DNA ligase and cloning, so which will help us to bring the genetically modified crops and also which will help in exploring the genomic to the next level, that is the genomics and proteomics. So here we have the presentation objectives, and first objectives. So like define plant biotechnology. So in case, what is plant biotechnology? We should know that, and we should know the importance of plants and their benefits to the humankind. That also has to be understood by the scientists or who were studying the plant biotechnology subject. And if you see the next point, identify the methods of gene transfer. So this is the one of the important thing. And then we will have explain the polymerase chain reaction and identify some genetically engineered crops. Then you will have contribution of transgenic plants to human health and nutrition. And finally, we should obtain some knowledge about the genomics and proteomics. The first one, what is plant biotechnology? So this plant biotechnology, which is basically the application of scientific methods to manipulate living cells or organisms for practical uses. The next thing, the biological phenomena for copying and manufacturing various kinds of useful substances. And also it will help us in controlled use of biological agents such as microorganisms or cellular components for beneficial use. And next one, we have historical overview of plant biotechnology. So where we have Matthias Sheldon and Theodore Swan. So these are the two scientists. They worked on the concept of cell theory in 19th century. And we have the father of plant biotechnology, that is Gottlieb Haberland. He is the one who brought a lot of techniques into the plant biotechnology and plant tissue culture. He is basically a German botanist and proposed plant tissue culture. And also we have some more history along with it. In 1981, scientists at Ohio University proposed first transgenic animals by transferring genes from one to the another. And also in 1980, genetic engineering was first used. Methods of plant biotechnology. So here we have gene transfer, plasmids as a vectors, and restriction enzymes, DNA ligase, cloning, polymerase chain reaction, tissue culture. So these are the things we have seen it in already in other subjects also. So here we have the next one, we have gene transfer. In gene transfer, generally the insertion of unrelated genetic information in the form of DNA into some other unrelated cells. And then we have gene can be transferred between the species through the genetic engineering. And next point we have genes can be transferred into two methods. One is the viral mediated, another one is the direct gene transfer. So with the result of both these transfer methods, we can bring about new transgenic plants or animals or organisms, and also which will give this transgenic plants, which can be done through the vector mediated gene transfer, which is carried out by factor called agrobacterium mediated transformation. So this particular bacterium mainly used in plant transgenic plants. So if you want to generate a particular transgenic plants, so in that time, so you can go for agrobacterium mediated transformation. And next one, we have Agrobacterium tumefaciens. So this is the another soil-borne gram-negative bacterium. So this bacterium mainly, there are two mainly species, that is the Agrobacterium species, that is Agrobacterium tumefaciens. Another one is the Agrobacterium rhizogens. So these two organisms which will induce some sort of diseases. If you see the Agrobacterium tumefaciens, where you can induce crown cold disease. The another one, rhizogene, that induce the hairy root diseases. If you think about the Agrobacterium tumefaciens, this is the phytopathogen, and also it is treated as the nature's most effective plant genetic engineer. So this is the symptomatic diagram representation for gene transfer. So if you see, this is the particular vector molecule, that is the plant vector molecules, are the cells, Agrobacterium tumefaciens bacterium. So in that we have the PI plasmid, tumor inducing plasmids. So in this particular plasmids, we can able to edit by inserting our gene of interest. Once we able to introduce our gene, and then which will transform again into the host cells, that is the bacterium cells, and then which will get replicated again and again. It will started to produce, and where we can able to see that results in our cells, and also from the cells we can able to grow the entire plant. So this is the another important thing, plasmids as a vector. The vector is an agent that carries gene from one to the another organisms. And also these plasmids will basically self-replicating circular DNA molecule in bacteria that are separates from and also smaller than the bacterial chromosomes, which carries plant genes. So, and also here we have the some basic vectors which is already available for transgenic plants. First one is the tobacco mosaic virus. Another one is the cauliflower mosaic virus. The first one, that is that diagram they have given here, tobacco mosaic virus. So here you can able to see the structure, how it is looks like, either it is a helical and what arrangement the nucleic acid which is made, and what is the nature of capsid. All the details they have provided here. So here you can able to see that in center place you will have the center core and RNA molecule as a ring, and then you have the capsomers as the capsid. So this is the another important virus vector, that is the cauliflower mosaic virus. That is genome of cauliflower mosaic virus they have provided here, and they have not given the detailed structure like earlier one. So here we have the genomic of cauliflower mosaic virus. So here you can able to see that different genes are present in this virus. The next one, restriction enzymes and DNA ligase. Restriction enzymes and DNA ligase are used to make recombinant DNA, and restriction enzyme is a DNA cutting enzyme that recognizes specific site in DNA. And also it can able to produce the sticky end or blend in nature. The most common restriction enzyme is the ECO1, and DNA ligase is the joining. This is the DNA ligase is the joining enzyme. So here you can able to see that the first steps, the steps which is followed in the transgenic plants are the recombinant plasmid preparation. The first one is the DNA fragments with different restriction enzymes. Once it is made, that is the cutting of DNA, and then the joining part, that is the ligation process through different DNA ligase enzymes. And once you have completed your ligation process, and it can be continuously produced, or the cells will continuously divided one by one. So in that case, we'll be getting the clones. Okay. So this cloning is the process or the process of genetically producing exact copy of genes or cell or the particular entire organisms. The cloning produces multiple copies of recombinant DNA. A clone is an exact copy of gene or a cell or the organisms. So here you can able to see that the another important point here, recombinant DNA involves modifying pre-existing genes to new genes. And also there are most common used plasmids are available for different types of cloning. That one is the PBR322 and P18. Even we have artificial chromosomes. So that also will be considered for cloning purposes. So here we have four major steps involved in cloning. That is the first one, we have to isolate the gene of interest, and also we have to select the particular vectors for transgenic plants or cloning purpose. And the second step, which will be followed as a ligations. Once it is followed, then you will go for transfection. And then once the transfection is over, and you will be going to select the recombinant or not non-recombinant. So that you will be selecting the recombinants only we need to take it for our cloning purpose. So here we have the another diagrammatic representation. The first one, like here we have cloning into a plasmids, like the plasmids. Here we have PBR322 or packet in any plasmids, and foreign DNA, that is the gene of interest. So in this particular gene of interest, that is in foreign DNA, we will select a particular region and we will restrict with the eco-hormone enzymes, and then both strands, that is the vector molecule and foreign DNA, both will be restricted with the same enzyme, eco-hormone enzyme, and then we will go for ligations. So this is the ligation process, and once it is ligation is happened, and then it will become a hybrid molecules, and from this you can able to obtain the recombinant DNA molecule. So this recombinant DNA molecule will be inserted into the host cells, and this host cells will start to continuously produce these vectors, and it will get expressed. In another case, so this host cells again will be putting into the medium, and where we can able to generate a lot of clones, and from the clones we can able to extract our gene of product, and from the product we can purify the product, will be purified, and then we will use it for commercial applications or healthcare industries, or based on different industry we will use that. This is the another diagrammatic representation for gene cloning. That is the bacterium. Again, we have a bacterial cells and plasmids, and gene of interest, which have been taken from another organisms, and restrictions which is happening, and again ligations, and similarly like you will have the transfections into the bacterial cells, and which will be getting expressed. So this is the another important thing, that is the method, polymerase chain reaction. The polymerase chain reaction, that is the clones DNA without using a cells. Okay. So if you see the cloning method and the polymerase chain reaction, we can easily compare the cloning method, which needs the alive cells to generate a lot of multiple copies, whereas the polymerase chain reaction, which doesn't need any alive organisms to multiple the gene products. And also, if you see the PCR, this PCR takes place in a small tube, and then there you will have the small amount of DNA, and also you will have the primers, and also the enzymes, and also the buffers and DNTPs. So these are the few elements will be available within that small tube, and then the process will be initially taken up one by step, and then finally you will have the multiple copy of particular gene. So here you can able to see this. So this is the DNA molecule. So this DNA molecule, if it is undergoes for PCR method, and where there are like initiation, elongation, and termination. So these are the thing that is in the way of, that is the denaturation. The first step will be the denaturing and annealing, and then finally you'll be having elongation and also termination. So these terms will be used in the PCR method. So if you give us double stranded DNA, so this double stranded DNA will be denatured, and from the denatured strand, the primer will attach to it, and the other DNTTPs or other molecule will help the primers to add more nucleotides along with the primer or the complementary to the DNA molecule with the help of different enzymes, DNA polymerase enzymes. So at the end, you will get the lot of DNA molecule from single DNA template. So here we have the methods of insertion, inserting a clone genes into the plant cells. So we have to select, first of all, we have to select the gene of interest, and if the gene of interest is selected, and then we have to select for the vector. Once we selected the vector molecule, and if we want to go into the biological way, so we have to select the host cells, and before doing all these things, we have to do restriction, ligation, and transformation into the host cells, and then it will get expressed. So this is the laboratory process, whereas the PCR method, where you will need to have only the machines. So in that we have to add components. So these components will mix together and will start to produce appropriate components. So this particular methods of inserting a gene clones are the clone genes into your plant cells, which can be either derived from the biological sources or from the mission, that is the instrument from the PCR mission. The first thing, like from that we can able to obtain. So any one of the genes or the DNA molecule can be taken, and from this molecule can be try to put into the plant cells. So for that, there are methods for transferring this clone gene into the plant cells. That is the gene gun, electroporation, microinjection, liposomes. The first one, gene gun. In gene gun, this is the ballistic particle which is delivering a system originally designed for plant transformation. So this gene gun will have a particle. So this particle will be coated with the cells, and these cells will be high phosphate, which will be injected into the target sites. So here you can able to see that gene gun barrel. So this is the barrel from where the particle will be there. That is cells along with the particle, gold particles are the different coated particles will be there. So this particle will be directed towards the target plant cells. So this is the another method, electroporation. So in electroporation is the microbiological technique in which electrical field is applied in a cell in order to increase the permeability of membrane. So if we increase the permeability of membrane, we can able to push the vector molecule or the recombinant vector into the cells. So here you can able to see that before electric pulse, so the membrane will be intact bilayer, whereas if you after applying the electric pulses, so which will look like this. Okay, so you can able to have the pore, and this through this pore you can generate, or you can try to put your vector molecule into the cell. This is the another method for gene transfer, microinjection. Microinjection, it is uses of micropipette to inject DNA into the cells. So here you can able to see that micropipette, so which contains the cells, the vector molecule, and so this recombinant vector molecule will precisely deliver into the cells. exact cells. So that is possible with using different microscope. So this is the another gene transfer method, liposomes. Liposomes are the small spheres made up of liquid molecules that can fuse with the plasma membrane. So once it is fused, and through that it will uptake by the cell membrane. Once it is uptaken, and which will leads to the nucleus, and there it will interact, and again it will start to replicate. So this is the other important thing, tissue culture. So we have seen the methods, and we are going to look into the tissue culture, that is the plant oriented. So this tissue culture, the plant cell, if you take any cell from plant, so it can be able to regenerate entire plant. Okay. So if you are doing it outside the normal environment, so in that case, the entire process or the generation of plants will be calling it as the tissue culture. And the cells are the plant, that is the source plant or their sample will started to form mass of undifferentiated cells. So that will be called it as the callus. So this callus can be able to bring down entire plant or the whole plant into alive. So here we have two types of tissue culture. First one is the anther culture, another one is the meristem culture. In anther culture, so you can take up the anthers, and this anther can be cultured in nutrient medium, and this nutrient medium will start to give a lot of nourishment to the cells, and from the callus generation. So here you can able to see the callus generation from the anthers, and this callus, that is the differentiated cells, can give a rise to the differentiated plants at the end. This is the haploid plant. So and also here you can see embryoid model. So this embryoid model also can able to give the haploid plantlet. Another culture, that is the plant culture method, meristem culture. So here we can able to take the meristem, that is the tip of or actively growing part has been taken, and from there we will start to do our culturing techniques. And once we culture that, and this culture can give rise to the new plant. So this will be taken into the field, and we will be putting there, and it will give rise to the entire plant. So here comes the genetic engineering. So we have seen the techniques and methods and even tissue culture. Now the important part, genetic engineering. So genetic engineering, which transfers a gene from varieties of organisms into the plant. So if I want to introduce a animal protein into your plants, so that is possible by this genetic engineering tools. Genetic engineering has made pest resistant plants, and also which makes more protective plants, and also which can make the plants resistant to pest and viruses. Against viruses, the plants can withstand against viruses, fungi, and also the plants can be able to produce the nutrition value protein molecules, and you can able to make different genetic testing through these plants, and also you can give a lot of hybrid plants or the nutrition value plants through this genetic engineering. So this genetic engineering comprises of artificial manipulation, modification, recombination of DNA or other nucleic acid molecules. Genetic engineering also made more plants which is resistant to harsh soil condition and pest. And also these plants can overproduce particular components or the particular product. And transgenic plants can increase the crop productions, and transgenic plants can be able to contribute to human health and nutrition. And we have these plants, transgenic plants, again increase the crop production, and finally, so this can give a rise to the human health and nutrition. So transgenic plants, here we have the transgenic plants. This benefits of transgenic plants to humans, if you see medicinal benefits. So it can give lot of components. So these components which is needed for affected persons or who's in ill. And then the transgenic plants can give rise to lot of industrial benefited products, and through that the industries can be rely upon plant industry also, and also agricultural benefits. And you have transgenic plants which improves poor access of food. So if the people who were living in poor country, so this transgenic plants which will be grown very short duration, and it can able to produce larger quantity, so we can eradicate the poor condition in particular countries. The advantage of genetically modified crops, if you see that the nutrition content can be improved here. And then we have genetically modified foods can have longer shelf life. That is one more important point. And we receive medical benefits from the genetically modified crops. And also we can create foods that are more appealing to eat. So nowadays the appealing is very important. For many are believing this appealing also will make the people to attract to that food. And GM foods also very easier to transfer from one to the another location. Even if you think the conventional plant materials or the plant products which can be easily decayed within a short duration due to its very shorter shell life. And disadvantage. So if you see the disadvantage here, the genetically modified crops may cause antibiotic resistance. Genes can go into different plant species. So that's also very biggest problem we have it. And the independent research is not allowed. So these are the few areas we have to consider whenever we are going for genetically modified crops. So these are the few brief history on the first genetically modified crops. So you can able to see that the normal tomato and with genetically modified tomato. So here you can able to see that the entire content which is completely different. So this particular plant which was produced in 1994, the company name called Calgene. This is another important transgenic product, golden rice. So this rice which has the benefit which can able to synthesize beta carotene. So this beta carotene, which is the part of the dietary vitamin A, so which can able to eradicate or which can able to control the blindness in childhood. And also this vitamin A, which is very essential for the children's for in developing ages. And it was done in Philippines in 1999. This is the another last thing, like from the entire lecture, genomics and proteomics from transgenic plants are plant biotechnology, so which can explore further into genomics, plant genomics or plant proteomics. The genomics, which is the science which determines the nucleotide sequence of whole genome, so that is the genome. If you see the proteomics are the collection of proteins from entire organism will be detailedly studied in proteomics. So this expressed molecules will be studied in proteomics. Without expressed DNA or genomic DNA molecule will be studied in genomic studies. So here you can able to see that the first picture. So this picture which contains chromosomes, and from this chromosome, if you stretch it or if you started to look more deeper, and where you will have the genes. So in another picture, you can able to see this. So this is how the DNA molecule is within the DNA molecule, the nucleotides are arranged respectively. So this is the protein arrangement. So we have here some more basic elements like oxygen, carbons, nitrogen, amino acid side chain, and hydrogen. So these are the reference I have followed for this